Next one, after cell envelope, next one is the surface appendages. Surface appendages. Surface appendages. So these surface appendages are, for our convenience of study, we can divide them as two types: role in motility, role in motility, no role in motility. Okay, surface appendages, for our convenience, best can be studied by classifying them as two types: role in motility, no role in motility. So role in motility, the surface appendage that involve in role in motility is flagella. Okay, so flagella which involves in a motility, that means here helps in a locomotion. Motility or locomotion, role in motility or locomotion. So that is flagella. This flagella is made up of flagellin protein. Flagella is made up of flagellin protein. The flagella arises from basal body. So the flagella is made up of flagellin protein, arises from basal body, and 9+2 arrangement is absent. 9+2 arrangement is absent. Flagella 9+2 arrangement is in flagella 9+2 arrangement is absent, made up of with flagellin protein, arises from basal body, mainly helps in locomotion. Next one is no role in mobility. So the structures that are involved in no role in mobility are, first one is pili. Okay, so this pili are long. Okay, long structures and made up of pillin protein. Main function is to form conjugation bridge during conjugation. One means of sexual reproduction in bacteria, conjugation. Pili helps to form conjugation bridge during conjugation. Hence pili are called as sex pili. These are called as sex pili. Okay, these are called as sex pili. So pili, long structures made up of pillin protein, helps to form conjugation bridge during conjugation, hence called as sex pili. Okay, next one is fimbriae. Next one is fimbriae. So these are short structures. These are short structures, okay, and made up of fimbrine protein, okay. So these are called as infectious pili, called as infectious pili because they helps in attachment to substratum or host body, helps in attachment to the substratum or host body. So these are fimbriae, short structures made up of fimbrine protein. These are called as infectious pili because they help in attachment to the substratum, or we can call it as a host body, whatever it may be, the host. So this is called as fimbriae. So like this, the surface appendages are classified in order to study in detail. These are classified into two types: rolling, that involve in rolling mobility or locomotion, and has no role in mobility or locomotion. So which plays role in locomotion? The structure is flagella, made up of flagellin protein, arises from basal body. 9+2 arrangement is absent. And next, no role in mobility. Those are pili and fimbriae. Long structures. Pili are long structures made up of pillin protein, helps in to form conjugation bridge during conjugation. Hence, they are also called as sex pili. Next one is the structure fimbriae. Short structures made up of fimbrine protein. These are called as infectious pili. Helps in attachment to the substratum or host body. Okay, so this is about the surface appendages. Next, cytoplasmic contents. Next one is cytoplasmic contents. Okay, cytoplasmic contents. The first structure is mesosome. In cytoplasmic contents, first structure is mesosome. So the mesosome is nothing but infoldings of plasma membrane is called as mesosome. Infoldings of plasma membrane is called as mesosome. Okay, so the main function of mesosome increases surface area, electron transport chain occurs. That means in respiration, the last step, electron transport chain, where ATP where NADPH and FADH energy releasing products are formed here. So those are that chain process, electron transport chain process takes place here in the mesosome. Helps in DNA replication. Okay, helps in during cell division. Helps in DNA replication, helps in cell division, increases surface area, electron transport chain occurs in the mesosome. Okay, so this is a like mitochondria, where in eukaryotic cell, where this process takes place mainly in the eukaryotic cell in mitochondria. Here in prokaryotic cell like bacteria, the process takes place in mesosome. Okay, so that is the first structure called as mesosome. And second structure is nucleoid, or which is circular double stranded DNA. Okay, main genome is called as nucleoid, which is circular double stranded DNA, which lacks histone proteins. That means histone proteins are absent. Circular double stranded DNA, main DNA, which lacks histone proteins. Okay, which lacks histone proteins. Next, third one is plasmid. Extracellular double stranded DNA are called as plasmids. Extracellular double stranded DNA are called as plasmids. Extracellular double stranded DNA are called as plasmids. Okay, these are self autonomous, okay, replicating double stranded DNA structures. Okay, automatically they will replicate, self autonomous replicating double stranded structures. These are again further classified as F plasmids, R plasmids, Vir plasmids. Okay, F plasmids role is during that is transfer of genetic material. R plasmids means their role is resistance, provides resistance. Okay, Vir plasmids. Okay, Vir plasmids. So like that, different types of plasmids are there. Okay, that we will discuss in biotechnological applications classes. Okay, so that is about plasmids. And apart from these plasmids, next one is the fourth one, ribosomes. Ribosomes. Okay, here in bacteria 70s type of ribosomes are present. 70s type of ribosomes are present. Helps in protein synthesis. Helps in protein synthesis. Okay, helps in protein synthesis. Next, fifth one is inclusion bodies. Non-membranous structures. Inclusion bodies are non-membranous structures. Mainly helps in storage of food. Mainly helps in storage of food in the form of phosphate granules. Helps in storage of food in the form of phosphate granules. Okay, helps in storage of food. Inclusion bodies, non-membranous structures, helps in storage of food. Maybe some phosphate granules. Okay, cyanophycean granules. Okay, in cyanobacteria. So like that, they helps in storage of food. Okay, in bacteria. Apart from this, gas vacuoles. Gas vacuoles are present in cyanobacteria. So main role is to give buoyancy in order to float on the water. Okay, main role is to give buoyancy. That means with the help of these gas vacuoles, due to the presence of these gas vacuoles, those cyanobacteria can float on water. So that is about gas vacuoles. So these 5, 1, 2, mesosomes, nucleoid, plasmid, ribosomes, inclusion bodies, and gas vacuoles. These are the contents of cytoplasm of the cytoplasmic contents, okay, of the bacteria. Okay, this is a detailed structure about bacteria. Okay, further we will discuss in our lecture 2 about bacteria. Further we will discuss in detail further contents like gram staining, gram positive, gram negative bacteria, and reproduction in bacteria, how it takes place. All these things we will discuss in lecture 2. Thank you, one and all.